Nitrate is a bladder cancer inducer and also has been shown to cause cytotoxicity in primary bladder cells. On the other hand, metformin is having an opposite effect. It protects against nitrate cytotoxicity and also protects against the nitrate induction to cancer. So it's anti-carcinogen as well. And metformin works through AMPK. AMPK is the AMP kinase protein and activated protein. This is a body or the energy sensor of the body. It basically balances the energy. So metformin can act through this and also can act independent through other pathways and can induce its protection in other and do its function through other avenues. So we'll talk about this. But first, I want to tell you about the talk. It's divided into 4 parts. I'll talk first about the introduction, and I will let you know about a little bit about the prevalence of bladder cancer and the current treatment that is available. Then I'm going to go into the background, and I'm going to talk about nitrate, metformin, and AMPK, the interplay of those 3 with respect to bladder cancer. And then we'll talk about the objectives of my study and the experimental design and how those experimental design would answer the questions that wanted to be investigated with the objectives. And then finally, I will discuss the results of my research. So the prevalence of bladder cancer, it is considered the 4th most common cancer in the United States. So currently there is about 587,000 patients living with bladder cancer. And this year, in 2018, there is 81,000 new expected cases, and there is 17,000 deaths. However, the worldwide prevalence of the disease is far more higher, around 500,000 yearly expected cases. Now, there is currently no cure for bladder cancer, and there is no preventive measures. However, there is really an invasive treatment. This treatment for bladder cancer is composed of 3 different types of treatment. The first one, and all 3 of them are really have side effects, as you can see. The first type is bladder cancer surgery. So that is the where they remove the bladder cancer completely or partially. And usually they also follow up with intravesical and chemotherapy. These are very potent chemicals, like this chemical here, mitomycin, which really causes a lot of side effects, including pulmonary fibrosis. That's the scarring of the hair sacs, destruction of red blood cells, tissue damage, hair loss, and also risk of infection. Radiation also follows the surgery here, but the radiation also has its own side effects, which is problems holding urine and radiation lesions causing blood in the urine and so on and so forth. One thing I needed to mention about the surgery, of course, after surgery there's bleeding and pain, but also there is another procedure that follows because that cancer usually reoccurs again, and by reoccurring again, they have to now burn the tumor. That's a very painful treatment. So there is a lot of environmental triggers that will trigger bladder cancer. One of the primary cause or the primary triggers is nitrate. According to the American Cancer Society, they identified nitrate to be one of the main or the primary causes of cancer. And also the World Health Organization in 2015 announced that it is a carcinogen group 1. So how does nitrate cause this effect? It does that through its derivatives. It breaks down from nitrate to nitrite, and nitrite becomes further broken down to nitric oxide. Then nitric oxide forms what is known as the N-nitrosyl compounds. These N-nitrosyl compounds are extremely reactive, and they are considered to be carcinogens. Okay, all of that nitrate and all of the derivatives of nitrate eventually accumulate in the bladder. But the reason it accumulates in the bladder is due to the fate of the dietary nitrate. So what's that fate? It's basically taking the nitrate, taking it out by the ingestion. In the oral cavity, there is bacteria that would break down nitrate to nitrite. Nitrite will be further broken down in the stomach because of the acid, and it will be converted to nitric oxide. And then from nitric oxide, you also form the N-nitrosyl compound. All of these will deposit here in the bladder. Once they deposit in the bladder, that's where the problem starts. They become harmful. So how they become harmful? Like I said earlier, they would form highly reactive species, like this particular species right here is the N-nitrosylamine. And what does it do? It actually translocates into the cell nucleus and starts its damaging the DNA, either by DNA methylation, or it could cause double-strand breaks, and so on and so forth. So I think now is the time I can introduce to you metformin, because metformin is going to be able to take out this effect, be protected against these species. So what is metformin? Metformin is actually a diabetic drug. It's for diabetes. It's already an FDA-approved drug, and for the treatment of diabetes. But the key function of metformin is to lower the glucose levels and increase the sensitivity of insulin. And this is a key one. There's also a lot of studies that show that there's a relationship between metformin, diabetes, and bladder cancer. So a lot of the demographic studies have shown that 80%.
Maybe it's low on the.
Can I continue? Okay. So 80% of diabetic patients have this condition. This. How can I continue?
Go on the other side.
Is that better? Yeah. So 80% of these patients develop this condition here that's known as diabetic bladder dysfunction. And those 40% of those that develop this diabetic bladder dysfunction actually have high risk of bladder cancer. Metformin, on the other hand, shows all these good effects. It shows anti-carcinogen effects through demographic studies and also lab studies. It also shows anti-cytotoxicity in primary cells. And it has also roles where it can remove the harmful effects of reactive oxygen species and reactive nitrogen species. So it removes the damage effects of free radicals. And also metformin has been studied extensively that it acts through these adenosine monophosphate activated protein kinase, the AMPK activated protein kinase. And there is a particular pathway that we will talk in details that it acts through, and it supports cell survival. So AMPK, like I said earlier, it's a master switch, or it's an energy sensor. What it does, it senses the energy, and when there's an imbalance, or there's, tries to bring that back to equilibrium by doing two things. It tries to increase the catabolic pathways. These catabolic pathways basically produce ATP, and try to inhibit or lower the anabolic, or inhibit this anabolic pathways, which consumes, and you guys understand synthesis of proteins and things like that would fall under these pathways. AMP regulates the oncogenic pathway PI3K. So this is the most famous pathway that AMPK works through. And this pathway regulates cellular survival and proliferation. But, however, depending on the cellular context, it depends how it regulates it. If the cellular context has certain factors, it may positively regulate it. If not, then it can negatively regulate it. If it positively regulates this pathway, then it will support cell survival. If it negatively regulates it, then it will initiate apoptosis and cell death, okay? So that's what I mean by the dual rule. So let's talk a little bit more about this dual rule of AMPK. That dual rule means that in certain environmental factors or context of the cell, like metabolism or activation intensity and the timing of activation, certain subunits of the protein, but mostly the microenvironment and the metabolism, AMPK can either be a tumor suppressor or it can be a proliferation promoter. So it can either support the cell survival or it can kill the cell. For example, like in this research, I have used normal primary cells and a cancer cell. So there are quite some differences between the two as far as metabolism and the microenvironment. So, for example, normal cells rely mostly on energy. They utilize oxygen. Cancer cells, they rely more on glucose. You have also pH. There's a neutral pH for normal cells. There is an acidic environment favorable to cancer cells. Also on the DNA or the chromosomal stability. Instability with the cancer cell, there's stability with the normal cell. And also cell cycle division. So we know that the cell cycle in normal cells are regulated. However, in the cancer cell, it escapes that cell cycle and it divides uncontrollably. So, and also one more thing, difference between them is cell-to-cell inhibition, contact growth inhibition. Cancer cells lose that cell-to-cell contact inhibition and they start to proliferate uncontrollably. Okay, so what is the, so introduced metformin and AMPK, so maybe now I should talk about how metformin interact with AMPK. So basically, metformin can have protective measures through its interaction with AMPK. That's one pathway, and that pathway is a dependent, AMPK-dependent pathway. Whereas this other pathway is this is a dependent pathway on AMPK. This is an independent. So this one, it can do it by itself without having to work with AMPK. In this pathway, it would activate this liver kinase protein. And when it activates this liver kinase protein, in turn, it activates AMPK. AMPK will activate this oncogenic pathway that if it activates it, it will support cell survival. If it inhibits it, it will cause apoptosis. On the other hand, metformin by itself can inhibit mitochondrial complex 1 inside of the mitochondria. It can also inhibit the NADPH oxidase, this enzyme. Both inhibition of those two would prevent the NOCs, or the N-nitrosyl compounds, from forming. Another thing it can do, it can initiate DNA repair mechanism by also activating this particular pathway. And then finally, in the inner mitochondrial membrane, there is this protein that is known as permeable transition pore. This protein forms when there is a radical or free radical insult. What it does, it makes the inner mitochondrial membrane more permeable, so the release of cytochrome C and other factors will initiate apoptosis. Okay? So I want to talk a little bit about this pathway here and explain that a little bit. Metformin, going through this pathway, in the case of, for example, activating this pathway, which supports cell survival, it will be through the AKT. AKT is the one that's responsible for it. So AKT is going to do one of these, is going to do these three things basically. It's going to activate mTOR, okay, rapamycin protein. That will lead to protein synthesis, okay? However, these are the really two important ones, because when ACT inhibits BAX, BAX will not be able to initiate apoptosis, so it will prevent apoptosis. However, when it inhibits FOXO, this is another protein that is a tumor suppressor. So when you inhibit FOXO, you will prevent growth inhibition. Okay. So let's look at AKT and more. Let's go inside and look at a more specific pathway. A more specific pathway is here again. Metformin activates LKB1 and phosphorylates AMPK. So when AMPK is phosphorylated, now it's active. Then it will activate, depending on the cell context, it may activate or inhibit AKT. When it activates AKT, when AKT is active, it would inhibit FOXO, the tumor suppressor. So it won't be able to carry out its function. So it won't be able to activate those pro-apoptotic proteins that would otherwise form these pore formations in the outer membrane of the mitochondria and cause the release of cytochrome C. The release of cytochrome C is required for the assembly of this complex known as the apoptosome. The apoptosome is basically what activates caspases, and caspases, these are the death molecules. When they're activated, they will initiate apoptosis. Okay? So, so this is the dependent pathway on AMPK that we just discussed. The independent pathway would be, like mentioned earlier, inhibition of NADPH, inhibition of complex 1. Okay, when it inhibits them, it reduces ROS and RNS generation, and then you will reduce the insult. Also, through this particular pathway, it can initiate DNA repair. And then also the last one, which is the inner mitochondrial membrane with the formation of the PTP. It will inhibit its formation, so the cytochrome C and calcium cannot escape and form this apoptosome, which will activate cascade. Okay? So at this point, I would like to talk about the objectives. So what is the objectives of this research? The objective of the research is to investigate the effect of metformin on primary bladder cells with or without nitrate and AMPK inhibitor. This is an AMPK inhibitor, dorsomorphin, which is also known as compound C. When you use it, it will stop AMPK from doing its function. It will inhibit it. So if I do that, what do I want to know? I want to know if metformin protects against nitrate cytotoxicity in human primary bladder cells, and also I want to know if it promotes bladder cancer cell death. Okay? So to achieve these objectives, this is the procedure in the lab that was taken. This is all cell culture procedure. Cells in liquid nitrogen are thawed, and then cell count is performed routinely, cultured in 25 centimeter square flasks, and this is the seeding density that starts with 3,000 to 5,000 centimeter cells per centimeter squared, and of course using the appropriate growth media for the cell type that is being grown. Then of course using the cell incubator, 37 Celsius at 5%. Once they reach 80% confluency, they either are passed or they are plated for experimentation. So once that happens, then you treat with the chemicals and the desired concentration, and then I use the trypan blue exclusion viability assay to determine percent viability of the cells with different concentrations. So what are the concentrations that are used? For metformin, 1, 5, and 10 millimolar. For sodium nitrate, 30 to 2,000 micromolar. And then I also use 10 micromolar AMPK inhibitor, compound C. And the endpoint measurements were at 24, 48, and 72 hours. Okay, so this is the first experiment, which the question that's asked here is that, does sodium nitrate causes any, if you treat primary bladder cells with sodium nitrate, what happens? Okay? And if you treat sodium nitrate with metformin, what happens? So this is a 24-hour, and as you can see, here is the increasing concentrations. It's shown here from 30 to 500. And here is the viability, and that's normalized percent control. And if you can see, there is a gradual cell death with increasing. Okay, so now what does NaNO3 does? It kills the cells, okay, with increasing concentrations. Okay, what about metformin? Well, an incubation of 48 hours with metformin showed no statistically significant change in the viability. So metformin does not hurt the cells, NOH hurts the cell. Okay, so what's the next step? What if we do a double treatment? So we treat this one and this one together, see what happens. Would this pattern stay or nothing happens, or would it be something in between, okay? Before that, I just want to show you this in another representation. Here is the decrease of the treatment with the sodium nitrate to the primary cells, and this is the metformin treatment, okay? So with the double treatment, here is 2 time points for the double treatment, 24 and 48 hours. Sodium nitrate alone, metformin alone, sodium nitrate and metformin. So this is simultaneously treated together, both of them, okay? So what do I get here? I get the lowest viability with sodium nitrate. And then I get the highest viability with metformin alone. And we saw that earlier. NaNO3 was inducing cell death, whereas metformin by itself did not have any change in viability. So what happens when we have the double treatment? Well, we have some rescue, but not to the full extent, but there is some rescue. And that shows that there is protection. Metformin does show protection. 48 hours will be the same, but there is a glaring difference between the two. And that difference is that NaNO3 here had more time to be incubated. This NaNO3 treatment alone had more time to kill the cells. So instead of 70, after another 24 hours, it's 48. So here, the double treatments pretty much stays the same, and metformin also doesn't change after 48 hours. But so, which shows that metformin is actually, for fact, is causing cell protection. Okay, this is that same experiment that I was just showed 24 hours and the 48 hours. This is sodium nitrate by itself, metformin. This is sodium nitrate and metformin. And you can see here that after 48 hours, more cell dies. Okay? Okay, so the next experiment is what happens if we treat one and then treat the other and then reverse that order. So now we're talking about the sequence of activation, or the activation sequence and the timing of event. Does one will inhibit the function of the other, or would they not independently do their job? So, and that would show also prevention. If you treat with metformin first and you get a good result, then that means you have a prevention effect, not just protection. Okay? So this is an experiment where the addition order is different. So the first one is just treating with NaNO3, that's the first one. And the second one with just metformin. And then the third one with compound C, which is the AMPK inhibitor. Here, metformin first, followed by sodium nitrate. And the other one is sodium nitrate, and then followed by metformin. And then treatment F and G, those last two treatments are exactly the same as this, with the exception that there's a pre-incubation with the AMPK inhibitor. Okay? So what happens? Lowest viability is from sodium nitrate. Metformin has the highest. Okay? We've seen that before. Compound C, which is the AMPK inhibitor, dorsomorphin, does not, it also doesn't affect the cell much. There's no statistical significance really between it and just metformin. Here, the double treatment viability went lower, where we can see that NaNO3 probably caused some cell death, but metformin was protective. And in this case, note, this is 24-hour and then 48 hours. So the total is 72 hours, okay? The same thing here. When I treat with this compound T, I see that the cells still have, some percent viability. It doesn't go all the way down, which basically infers that AMPK is a partial player in the, but not completely. There is other mechanism that metformin is acting through, not just through AMPK. So that is metformin treatment. This is AMPK inhibitor treatment. This is metformin first for 24 hours, and then 48 hours the NaNO3. Then here Na and sodium nitrate first, and then the metformin. So 24 hours, then 48. This is with the same exact, but for the pre-incubation of the AMPK inhibitor, and this is the NaNO3 by itself. Okay, so we can basically conclude, or before we conclude, actually, this last experiment is about the cancer cells. So I used the cancer bladder cells. All the other experimentation so far were in primary cells. This one is in cancer cells, and wanted to see what metformin would do. So treated with increasing concentration of 1,000 micromolar to even more than half of it was killed at 10,000, which is 10 millimolar metformin. So metformin killed bladder cancer, and there is a lot of studies showing this effect in other cell types. So what do we conclude with this? That metformin selectivity, treatment selectivity antagonized proliferation of bladder cancer cells while showing no effect on primary bladder cells. This cell selectivity suggested that metformin action in bladder cells is cell type specific. Switching the treatment order corresponds to the sequence of activation, which did not affect primary bladder cell viability. Metformin protected against nitrate cytotoxicity in human primary cells, and also it prevented cell growth from nitrate-induced cytotoxicity in human primary bladder cells. AMPK was partially involved in the metformin protection and prevention mechanism or measures. So a overall proposed mechanism would be that you treat with NaNO3, you start having free radical formation, reactive generation, cell death induction. You treat with metformin, it activates the mTOR PI3K pathway, that oncogenic pathway. When it's activated, it will support cell survival. And then you're going to also, metformin on the cellular level will inhibit the complex 1, the PTP inhibition in the inner mitochondrial membrane, and the NADPH oxidase. And then also it will initiate the DNA mechanism, the repair mechanism. Okay? And that's it. And I would like to, of course, thank you guys and thank my committee, my advisor, Dr. Fan, and my committee members, Dr. Yusufapour, Dr. Olofini, and Dr. Lian. Thank you. And also the Department of Environmental and Interdisciplinary Science for their support and the supplies to the supplies and funding. Also the Department of Chemistry and the Department of Pharmacy and Health Science. They supported a lot with the core facility where the experiments were done, and with also